Good day, class, and then welcome to the next episode of the microscope. Now, before you start watching the video, you have to follow the button and then like my page at the bio tutor, and then you also follow me on TikTok at the underscore bio underscore tutor, and then on YouTube the bio tutor. Now, today we will be talking about the types of microscope slide. The types of microscope slide. So we were talking about the parts of microscope, and then we made mention of the types of microscope slide. Now, when we heard of the microscope slide was when we were talking about where to place the specimen to be viewed under the microscope. And we said we place the slide on the stage. We place the slide on the stage. Therefore, what is a microscope slide? A microscope slide is a thin, flat piece of glass on which a specimen is mounted for observation under a microscope. A thin, flat piece of a glass on which a specimen is mounted for observation under a microscope. Now, somebody will ask, why are we using slide, or what are these slides used for? One, they provide a stable, flat surface for the specimen. Two, they allow the specimen to be positioned correctly under the objective lens. By now, everybody should know what objective lens is under the microscope. They protect the specimen and the microscope as well. And then they allow specimen to be stored and then reused. So these are some functions of the microscope slide, or why are slide used. Okay. Now there are two main types of the microscope slide. We have the dry mount and we have the wet mount. So we can say the dry mount slide and then the wet mount slide. Let's pick the dry mount slide and then let's talk about it. A dry mount slide is a microscope slide on which a dry specimen or a specimen in a dry medium is mounted without liquid. We can go to the market and say, I want a dry fish, meaning that this fish has been placed under the sun, or we've applied heat to it, and then the moisture content in the fish has been drained. Therefore, we don't have any interference with water. So with this type of microscope slide, which is the dry mount slide, the name itself should even define the slide. Meaning here, there is no presence of water, or there is no interference of water. So a dry mount is a microscope slide on which a dry specimen or a specimen in a dry medium is mounted without liquid. What are the characteristics of a dry mount? The specimen is placed directly on the slide. This specimen is placed directly on the slide. No liquid is added, and a cover slip may or may not be used. The specimen also remain dry. So in this position, everything is dry. And then a cover slip may or may not be used. Like, let's say we want to find the internal features of a cockroach, and then we place a cockroach on the cover slip, and then we mount it on the stage. Here, we place the cockroach on the microscope slide, and then we mount it on the stage. Here, the cockroach does not need any cover slip. Okay, so it may be used some way or the other. Some specimen will require a cover slip because maybe they will be lighter in weight, and then when there is a cover slip, it can cover it so that it will not be blown off. But the organism mounted on the microscope slide is dry. Example of specimen suitable for dry mount. We have the pollen grains, we have insect wings, we have hairs, we have fibers, we have dust particles, we have thin section of plant tissues. Let's look at some advantages of a dry mount. Advantages of a dry mount. Quick and easy to prepare. Dry mount is quick and easy to prepare. Now, if we want to investigate about the hair of an organism, the only thing we need is the specimen, which is the hair, and then we put it on the slide. Okay, so it is quick and easy to prepare. Specimen can be observed immediately. No staining is required. Staining means adding some dye or color to the specimen which cannot be seen under the microscope well, so that the color will give the specimen a bit of visibility. Specimen can be stored for long period. So dry mounts, specimens that undergoes dry mount can be stored for a longer period. So if you go to the labs, you could see that we have a lot of specimen being stored. Maybe they are preserved in a liquid, but when it gets to observation under the microscope or observation, we just bring them out and then we dry them and then later we observe it. Okay. But most of them are dry. What are some disadvantages of the dry mount? Some specimen may be damaged or distorted. So you find a cockroach there, and then one leg of the cockroach has been taken away, or the wings of a cockroach is not available. So some of the specimen may be damaged or distorted. Image clarity may be reduced. Image clarity may be reduced since it has been stored for a longer period of time. The clarity of the image may be reduced, and then specimen may dry over time. Specimens may dry over time. Specimens may dry over time. Now let's look at the wet mount. We are going to type 2, the wet mount. The name wet should tell you where we are coming from, meaning that this type of slide, the organism or it is being mounted in a liquid medium. So a wet mount is a microscope slide on which a specimen is mounted in a liquid medium. And when we say liquid medium, it is usually water or staining solution. It is usually water or staining solution. Okay. Characteristics of wet mount. The specimen is placed on the slide. 1. 2. A drop of liquid, water or stain, the liquid medium, I said it could be water or a stain, is added. 3. A cover slip is placed on the specimen. And then 4. The specimen is surrounded by liquid. The specimen is surrounded by liquid. Examples of specimen suitable for wet mount. Examples of specimen suitable for wet mount. We have 1, the living cells and microorganisms. So sometimes there could be an outbreak of disease, and then especially with the microbiologists, they will be saying that they want to research on the outbreak of disease. It could be a riverborne disease. Therefore, the source of this disease is a river. Therefore, they have to go and fetch water. Now they go and fetch water. There are microorganisms in the water. Micro means these things or these organisms cannot be seen with the eye. Therefore, it needs the microscope. So, microorganisms. We have living cells. We have onion epidermal cell. We have the cheek cell. We have bacteria. We have algae. Some people can say alga. We have the small aquatic organisms as well. So all these things are organisms that can be or specimens suitable for the wet mount. Now, what is the advantage of the wet mount? Specimen can be observed in a more natural state. So here, in this process, the organism is not dead, and then it could be observed under more natural state. So you fetch the water, and then you try and then view the microorganisms in the water, and then you see living cells moving around. Okay. Living organisms can be observed. 3. Stains can be used to highlight structures. Stains can be used to highlight structures. So here, structures that cannot be seen with the eye, since there is a presence of a stain, the stain can be used to highlight structures. Okay. And then the image clarity is usually better than the dry mount. Since we are using a stain, the stain will give the organism a color or a good visibility. Let's look at the advantages of using the wet mount. It requires more preparation time. It needs a lot of time. And if you don't take care, what you want to observe, you will never observe it. Specimen may move or be damaged by the liquid. And then the next one, liquid may evaporate over time because of heat. And a specimen cannot be stored for long period unless sealed. So always with a wet mount, they are like how should I say, a temporary slide, and then the dry mounts are like a permanent slide. But you can make uh, uh, a specimen in a liquid medium a permanent slide by covering it with the cover slip and then gluing the cover slip. So anytime you want to observe, you can just go for it and then observe. Okay. Now, let's look at staining. 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 When we say staining, staining simply means you are making the specimen visible. So let's look at the definition for staining. Staining is the process of adding a colored dye. The dye is not DIE, but DYE to a specimen to make it more visible under the microscope. A process of adding a colored dye to a specimen to make it more visible under the microscope. Now, somebody will ask why staining is necessary. Why staining is necessary? Many biological, specimens are usually transparent or translucent. So when we say translucent, we can see through, but transparent, we can see through it easily. But translucent, it cannot be seen through easily. And then we have opaque objects. Opaque objects, does not allow light to pass through, so it is difficult to view through opaque objects. But translucent and then transparent, you can see through. One is more easily, and one is not easy. Okay. So without staining, these specimens are, very difficult or impossible to see under the microscope. So, like, let's say you peel onion, you will see some whitish cell around it, the onion epidermal cell. Yes, and then although you cannot see through it easily, but you can see through it, and then it is white. With this type of organism, if you want to observe the internal structures of this type of organism, you have to stain it. So stain add color and then contrast, making structures visible. Okay. So let's look at some common stain used in biology. We have a stain like iodine. We have a stain like iodine, and then the color of this stain is usually brown or yellow. And then it stains starch and then nuclei. It stains starch and then nuclei. But you have to note the safety around this stain. It is very poisonous, and humans or living organisms should not ingest. And ingestion. Ingestion is the intake of food or substance. Okay, so it is very poisonous, and then we do not ingest, and then it can stain skin and then clothing. So when iodine stains your skin and clothing, it won't go. Okay. We have another stain like the eosin. Eosin, the eosin. The eosin is usually pink or red in color, and then this stains protein and then cytoplasm. It is very poisonous, and we do not eat it. It also stains skin and then clothing. We have the methylene blue. The methylene blue. It is our normal usual blue we've been using, but this one is chemically prepared, and then it is very deep and thick. When it stains your shirt, oh God, it will take a lot of time before these things move away. Sometimes it does not even go. As its name suggests, methylene blue is blue. It is blue in color. And then it stains nuclei and then bacteria. It is very poisonous and do not ingest and then stain skin and clothing. Now let's look at some important safety precautions when using a stain. Never eat stains. You should never eat stains. Wear clothes you don't mind staining, because stain may be permanent. So it is better when you're going to the lab, you wear your lab coat, because with the lab coat, you don't mind staining it. Wash your hands after handling stains. Please wash your hands. Students, do not do this. It is advisable for you to wash your hand after handling stains. The next one, you ask your teacher for guidance on proper use of the stain. And then the last one, you use stains only in a well-ventilated areas. So you don't go to one corner where there is no ventilation and then no. Because remember, we are saying it is poisonous. So you use stain in a well-ventilated areas. So these are some important things I wanted you to know under the microscope. The types of microscope slide, and then the type of stains or staining we have. We will be looking at preparation of a wet mount slide or preparing a wet mount slide. Thank you for your time. And then don't forget to press on the like button and then forward this video and then share it as well. Don't forget to follow my page and then like my button as well. If you don't understand anything, I can pull my number out. It is 0556 474887. And then you can follow me as well on TikTok, which is underscore bio underscore tutor. And then on YouTube, we have the bio tutor. Thank you for your time and then have a wonderful day. Bye bye.